This presentation is on Your Inner Fish by Neil Shubin, by Victoria Marble, Zach Bastres, and Emma Gollob. The overall idea of the book is that this book connects parts of the human body and how they relate to other animals and common ancestors in a previously unknown way.
Paleontologist Neil Shubin starts by asking, how can we visualize events that happened millions, perhaps billions of years ago, beginning his journey of anatomical history and recalling his own personal experience towards discovering his inner fish when he helped illustrate the human body's 3.5 billion year history. After spent years digging in Pennsylvania, he decided to expand his searching and explored the Arctic Circle, particularly Ellesmere Island. He describes his journey to find the fossil Tiktaalik, meaning freshwater fish, which was unlike any modern fossil or record. It had a neck, shoulders, and bones that corresponded closely with the arm bones in land animals. As a result, its unique structure has made it to be an important part of our anatomical history. Shubin tied these discoveries back to Charles Darwin's theories that suggested that all animals with limbs shared a common ancestor that gave them this limb structure, going all the way back to fish fins. It is common knowledge that the modern fish fins look nothing like limbs, being composed of webbing and 4 bones arranged in a line. But the lungfish shows bones being connected to his shoulder, and the Eucenopteron goes further by having 1 bone connected to 2 in the fin. Tiktaalik displays many of these features, and Shubin discovered that it would have lived during the exact time period of the transition between water and land animals. Going further than the anatomical structure of these fossils, Shubin looked into the DNA of these animals to see how they corresponded with the modern man. Randy Dawn at the University of Chicago was looking into DNA of sharks and skates, specifically comparing the DNA recipe of shark fins to human hands. During this, Shubin was looking for evidence to why the pinky looks different from the thumb as a key to how genetic recipe controls hands. In the embryo, limbs develop into paddles, much like fins, that would eventually form into hands. This is where ZPA, or zone polarizing activity, would come into play. It is a tissue that controls the formation of the fingers, without this, the fingers would not form. This gave way to the testing of the sonic hedgehog gene, which is only active in chicken wings, which gives them the form they have. Dawn then began to look for this in sharks to see if they possess the same gene. Teeth can also give many clues to evolutionary structures in animals. Teeth are the easiest finds for paleontologists because of an easily preserved hard mineral called hydroxyapatite, which makes up most of the outer layer of the tooth. Mammals have a relatively complex teeth that are reserved for a given species. Reptile teeth are much less complex than mammal teeth, but due to the fossil record, there seems to be a direct line from the simple to the complex. Many animals' teeth fit together to help gain more nutrition, something called occlusion. During Shubin's discoveries, he was fascinated at the occlusion of the teeth of tiny mammals from over 190 million years ago. It was thought that only these mammals showed signs of occlusion, but Shubin found tiny reptiles called trithelodonts that showed these signs. Although trithelodonts are reptiles, they have a mammalian jaw, marking them as an intermediate stage between reptiles and mammals, just as the Tiktaalik was an intermediate stage between fish and land mammals.
Chapter 5, Getting Ahead. In the beginning of Chapter 5, Shubin is talking about how he studied the cranial nerves back in college. This in turn led to him thinking about the essence inside our own heads. Shubin then continues to talk about the inner chaos of our head. According to Shubin, our heads are made up of 3 fundamental parts: the plates, the blocks, and the rods. The plates cover our brain and they grow just like we do, larger and stronger as you get older. The blocks have many arteries and nerves that run throughout them, and the rods make up some of our bones, such as our jaws, ears, and throat. They break up and change shape to help us hear, chew, and swallow. It then moved into the essence of the embryo section, which talks about how fetuses go from having gills to then transferring into breathing and how those muscles change for each stage of development. The next section, called Our Inner Shark, talks about how humans and sharks are similar and how they developed. Chapter 6 covered the comparing of the embryos of fish and amphibians and how evolution worked for both of them. Then Shubin continued to talk about how Speckman wanted to find where the bodybuilding is and wanted to know if it was connected to certain parts of the embryo. The next section talks about how Speckman physically distorted embryos to learn how their tissues build bodies. Speckman ended up winning a Nobel Prize for his work on embryos. Chapter 7, Shubin talks about the differences between a group of cells as an individual and just a group. As you can see in the chart, there was a very long time where there was only single or very few multicelled organisms living and did not make up any body of anything. Shubin then continued to pose questions about how any of our organs stayed separated, say the eyes or from our head or our stomach from our intestines. He believed that the different ratios of different types of cells forced them to stay separated. In Chapter 8, Shubin starts to talk about how you can extract DNA without a lab or any fancy equipment. Everything in our body does is in our DNA, and Shubin talks about that and how our sense of smell allows us to discriminate between 5,000 to 10,000 different odors. Smelling is like a lock and key mechanism. The lock is the odor molecule and the key is the receptor to our nerve cells. Each receptor recognizes a different cell and then it sends signals to our brain. Species can lose their sense of smell by damaging receptors or by mutations through evolution.
In Chapter 9, Shubin discusses how human eyes are related to the eyes of other animals. Humans have 3 molecules called opsins that make up the cones and rods in the eyes that allow us to see color and black and white vision. Most animals and fish have only 2 molecules, and it shows that humans have developed another molecule from animal ancestors. It was discovered that PAX6 was the gene that is used for developing eyes, so scientists manipulated this same gene in animals. They found that this gene also coded for manipulations in eyes of other animals and that they could grow eyes in different parts of the bodies of the animals. It showed how related the genes in humans and other animals like fish are. In Chapter 10, Shubin discusses the ears of animals and how they relate to the ears of humans and how they relate to other animals. Humans have 3 ear bones. 2 came from jaws of fish. As the humans developed smaller jaws, those bones slowly went up into their head. 1 came from the gill arch of fish, and it shows how closely related fish are to humans with those 3 bones. Mammal-like reptilians bridge the gap in fossil records such as Tiktaalik, which showed the process of the bones moving from the gill arches and the jaws up into the ear. Gel in the inner ear, along with the hairs of the inner ear, are used for balance and are similar to the gels found in the pores of fish along their lateral line that tell them different movements in ocean current. Today in humans, we use them for balance, but it's the same kind of effect. PAX2 gene was found to be the gene for ears in humans along with other animals. Scientists found that it can be manipulated in animals and that different parts of the animal ear can be turned on and off. Jellyfish were shown to have PAX6 and 2 as a gene combo, which could have meant they were early ancestor of both humans and fish. In Chapter 11, Shubin discusses the meaning of it all. He does a lot of summary, and he talks about the law of everything, which is that every living thing on this planet had parents. Whether it was trees and they had 2 trees pollinate, or animals that had intercourse. He also discusses descent with modification, which allows for lineage to be traced back step by step as different modifications came up through evolution. He discusses that every illness has something to do with evolving from other things. Every illness in humans is because of something else, such as obesity, heart disease, and hemorrhoids were from hunting and gathering. As fish or in animals, we always used to move, and not moving has caused diseases like obesity and heart disease. Also, sitting for long periods of time causes the blood to pool, which causes hemorrhoids, which is not a natural thing for other animals. Choking and sleep apnea come from the modified gill arches of fishing that we use for talking. Since we use the same passage for talking and eating, or breathing and eating, it can be difficult at times and choking can occur. Hiccups are also from the signal from muscle contraction traveling longer than it did in the fish ancestors, which causes spasms. The fun fact is that the longest bout of nonstop hiccups was from 1992 or 1922 to 1990, continuously. And then, as the picture shows, with the nice yellow arrow, you can see the shark gonads, which the hernias in humans come from, because in humans the shark gonads aren't up in the chest; they're farther down, and as we've evolved, that has caused tissue weakness in muscles, which allows for hernias to occur.